Nice. There is a picture. Is it recording? Yeah. There is a picture that Divya had sent to the group that shows the different cells. There's secretions and what the secretion does. So we can start with the alpha cells, which secrete glucagon, which increases hepatic glucose, GLP-1, GLP-2, GIP. Then you have the beta cells, which secrete the insulin, which is used to reduce the blood glucose, the gastrin, the CCK, the secretin, urocortin. Okay, some of these I don't really think they're that. Some of the main things secreted. Amylin, vasopressin, and the oxytocin. Oxytocin. Okay, that's I've learned today. Then you have the delta cells, which secrete the somatostatin, which inhibits insulin and glucagon. As you know, also glucagon and insulin are also not produced at the same time. We have the other cells, the epithelium cells, which secrete the ghrelin, which inhibits insulin, giving you the satiety. We have the PP cells, secretes pancreatic polypeptide, which also inhibits insulin. So yeah, the main ones, alpha, beta, delta. I think I've seen that person in different exam papers, mainly focusing on the alpha, the beta, and the delta cells. So according to now what we know, the pancreatic islets secrete all the following hormones except somatostatin, insulin, glucagon, secretin. Interesting. Because based on that,
all of them are answers.
All of them are answers. Are they all hormones?
Good question.
Yeah. Yeah, based on that, they're all being produced. Secretin. Okay. Yeah, but I'm saying it's being produced by the beta cells. The point is it's not a hormone. It's an enzyme. Maybe. Let me see. Secretin is a hormone. But if you're going to go with the obvious, I'd also go with secretin because I said mainly they deal with alpha, beta, delta. Even though it's being produced, like, to be honest, I'd also go with C. Does anybody else have a different opinion? Okay, the C's have it. S cells in small intestines. Yeah, Fatima, I agree with that. I've seen according to the diagram that had been sent to the group, it's like there's some levels of secretin also being produced by the pancreas. But if you're going to with the major things produced by the pancreas, secreting, I guess not one of them. All the following are examples of bile pigments, except acrobilin, urobilin. Have we covered bile? Have we covered any of these discussions? Kama when you are covering biochemistry. I take that as a no. Let me look for those marks. Emma, if you have any, you can share. I'm trying to look for them. Okay, I'm not able to find them now. Maybe when I find them, I'll share it in the group. Anyway, yeah. So all the following are examples of band pigments except stachybotrylene. Oh, see. Nice. Somebody has shared it in the group, like a picture diagram. Seraphine, will you be able to explain it or expound upon it? Or somebody else can do it. The experts in the group can help. Patrick. That's how they see. Let me try to see how it's also happening. Input. Oh, nice. So stachybotrylene gives stool its brown color. Somebody has said in the group, the stachybotrylene gives stool its brown color. Urobilin gives urine its yellow color. Yeah, because it can be excreted both through the urine and through the stool. Okay, that's a nice addition. Based on the diagram tumetumia kwa group, we have the mesobilirubin, then there's a mesobilirubinogen, then it's converted to stachybilinogen, stachybilin, which is then excreted. Then you have the stachybilinogen, it's like it's taken back to the liver. Oh, okay. I can't remember exactly the pathway and its explanation at this moment, but I'll go back to it. So based on that, we have stachybilinogen is there, mesobilinogen is there, stachybilin is also there. It's like what's being taken out. So that's D. D-urobilinogen. Then we have urobilin. We have urobilin, oxidation, avoiding. Yeah. Okay. So we can move on to the next question. Dash is the end product of glycolysis under anaerobic conditions. That one is an obvious one. Some given assumptions to an example. In aerobic conditions, pyruvate is the end product, and in anaerobic conditions, you have the lactate. That's the final product. I think you've covered glycolysis in previous. Yeah. So in I think something else you should mention is the end, not the end products, like how many ATPs are produced, the net in glycolysis. Sorry for that too. I think we have a gross production of 4 ATPs, but since we use up 2 in the process, so I think it's a net of 2 ATPs, but I stand to be corrected. Yeah. So yeah, net of 2 ATPs are produced. And what about NADH? How many are produced? Oh my God, I'm losing my laptop. 2 NADH also. Okay. So for number 1, all the following are processes in liposomal protein synthesis except everywhere sounds familiar. All the following are processes in liposomal protein synthesis except have termination, elongation, initiation, transcription. That's what I'm saying. My internet today is not on its best behavior, so in case I get kicked out again. Yeah, sorry. Yeah. So what are the processes in liposomal protein synthesis? Help me out, because even me, I don't know. Somebody says T, which is transcription. Okay. Take us through the processes. Honor, kindly. Or anybody else. And it's been sent to the group and I'm missing it. Let me know of which it has.
Oh,
Yeah.
Janet, I'm not really sure.
Mhm.
Just reading from the photo that I sent, so there's the start codon and the stop codon. The start codon is usually IUG and the stop codon is UAG. So initiation starts when the transfer RNA and starts to bring the amino acid in the polypeptide chain to bind to the start codon on mRNA. So so transfer RNA will bring amino acids to bring to the start codon when it starts binding. That's the initiation process. That's what initiates the protein synthesis, followed by elongation. Now the transfer RNA brings amino acids one by one to add to the polypeptide chain. So elongation just means like it continuously adds to the transfer RNA, continues bringing the amino acids from the amino acid pool and pull to the start codon so that it can be produced continuing, like elongation is a continuous process. And then for termination now, termination is stopped by when we reach the stop codon. Release factor recognizes stop codon and translational complex dissociate and completed polypeptide is released. So yeah, so once we reach the stop codon, the release factor now will recognize the stop codon, and then now there will be dissociation of the completed polypeptide. They'll be released, and that's the end of that's the end of protein synthesis. I don't know if that makes it sense. That makes it simple. I'm just honestly reading from the diagram. So if anyone has a better explanation, please go on.
Ah. Hey, thanks for that, Dr. Nugal. I'm also seeing Stanley is also adding more information and saying the stop codons are three sequences. We have the UAA, the UAG, and the UGA. These are for the stop codons. Did you come up on the start codons? All right. Let's go. And so by that, we have the answer. We started initiation, elongation, and then termination. So the answer is transcription for this one. So I'm seeing, yeah, Dr. Lin and Dr. Turi wameongeza UAG, the start codon. So AUG, sorry. AUG, the start codon. Thank you so much. The more we learn. Yeah. The start codons code for methionine. Okay. Thank you for that. Yeah. All the following, I've read that one. All the following are properties of an electron, except we have electrons are involved in chemical bonding. An electron possesses a spin. Electrons is both a wave and a particle. An electron exhibits properties of a subatomic particle only. Okay, so we cannot like the first one. Electrons are involved in chemical bonding. That's true. An electron possesses a spin. That's also true. An electron is both a wave and a particle. A wave and a particle. I don't know about that one. Somebody can help me out. An electron exhibits properties of a subatomic particle only. So between C and D, I am stuck between the two. Dr. Sunshine says an electron exhibits properties of a subatomic particle only. That is false. So an electron is both a wave and a particle. So I have learned that today.
C is true.
Yes, okay, thank you. Thank you so much. So now you've learned properties of electrons. Number 23. Which of the following states of matter is the most compressed? Okay. That's the question. Which is the most compressed?
I think
Okay.
liquid.
Compressible.
I
Okay,
think
okay.
that more.
Solid, gas, liquid, melt. Dr. Chur says gas. I agree with her.
Yeah, I also agree.
Yeah, that's undisputed. We need to talk more about states of matter. That is properties. It's good. Number 24. Which of the following represents, I guess, a balanced equation between arsenic trioxide and cerium sulfates? Now let me tell you. The arsenic trioxide is 3. The chemist in the group, the pros, Dr. Bonaya. I know you know these ones. But we cannot do it together. Arsenic trioxide, itakuwa na valency of the elimination it has the valency of 3 plus.
Yeah.
The whole equation. It's not complete, it's not like the answers are not, some part has been cut off. That's what I'm saying.
Yes.
So arsenic trioxide and cerium sulfate, no, they're not complete. They're not complete. It's hard to answer that one. Okay. So which one? Which one of the following represents the Beer-Lambert's law in the absorption of electromagnetic radiation? Yeah, absorbance. Since you've used it all your life. Yes, I agree with Dr. Lin and Stanley. That is A. Is a concentration. Yeah, absorbance is the absorbance. A is the absorbance. Yeah, absorbance is. Somebody state for us the Beer-Lambert's law. I can remember that. The relationship between the absorbers, the concentration. A tough crowd, eh? The absorbance is directly proportional to the concentration. Moving on. Yes, thanks, Dr. Lin. And other information is linked to the group. That's the genetics part. Which of the following types of bonds exist in a diamond? We know diamond is mainly carbon, so yeah, I agree with C, covalent bonding. We go on.
Yes.
Okay. Which of the following radionuclides is useful in brain tumor diagnosis? Have the iodine, have graphite. What about the graphite? Oh, you're asking what bonds exist in a graphite. Is that what you're asking? I'm thinking that's what she's asking. He or she is asking. So what kind of bond exists in graphite? Graphite is gel carbon, so maybe not guess. Graphite still has covalent bonds. If that was your question, if I got it right, if that was the question. Both covalent and van der Waals.
Yeah, covalent and van der Waals.
Okay.
So the key difference between diamond and graphite is that they are all carbons, but carbon will have 4 covalent bonds. All its 4 bonds are covalent, but graphite has 3 covalent and then 1 that is van der Waals, making it slide over each other. Yeah.
Thank you, Daktari. So we go to radiopharmacy, which of the following radionuclides is useful in brain tumor diagnosis? Brain tumor diagnosis. For this question, I remember there's a time, I think there was a discussion where you guys were discussing the different types of is it radiochemical reactions or something like that? And I wasn't available and I won't go through it. So can somebody please remind us? Ama whoever gave the crash course can read again for those of us who were not there. We learn these things, but yeah. Okay, let me see if I'm able to give a crash course. I guess you can be able to give that crash course.
Crash course on what?
You remember these things you were learning in radio pharmacy for like the alpha emissions, the different kind of reactions. I don't remember what they were called.
Yeah, I think someone sent a very nice photo on the group. If it can be re-highlighted, I think that would be great.
Kindly.
Yeah. So there was the alpha whereby
Okay, yes, yeah.
Yeah.
Hey, go on.
The beta, it was emitting a 0, -1. What is it? 0, yeah, 0, -1. The gamma was 0, 0. It was just emitting a lot of energy. Then for positron, on the product side, we had a 0 and a 1. So it's the opposite of beta. Then we had electron capture. The electron now was on the side of the reactant.
Okay, let me stop you there. Try it again in a what way? Like sikuelewi when you say 0, 0.
Now the photo would help if someone can highlight the photo. So but okay, when I say 0, 0, I mean mass number and atomic number. So alpha decay is there'll be emission of a helium particle. So 4 and 2. Helium
Okay.
number of 4, atomic number of 2. Then for beta emission is it will have a mass number of 0 and a -1 as the atomic number. A mass number of 0 and -1 as the atomic number. Then for positron emission is the opposite. It will have a mass number of 0 and an atomic number of 1. A mass number of 0 and an atomic number of 1. Gamma is both a mass number of 0 and an atomic number of 0. Then electron capture is the unique one because the electron will be written on the reactant side because it's the compound, it's the nuclide capturing an electron. So 0 - 1, then you will balance the equation accordingly. I'm seeing Dr. Divya has resent the photo, so I think that, yeah.
It's a nice summary.
Yeah.
Thank you so much. So which of the following radionuclide is useful in brain tumor diagnosis? Do, 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 do. Okay, look. Somebody technician. Okay, so C. Why not all the others? Okay, so C is the answer for that question. Technician is useful in brain tumor diagnosis.
So on
Okay.
that, to add on is that
Yeah.
technetium is if you look at these PET scans, when it has to do with visualizing, you notice that most of them technetium is the radionuclide. But for when it comes to cancer, so as to test the metabolism of different area, because cancerous cell will have more metabolism, that is when we give FDG, fluorodeoxyglucose. But merely you can't go wrong with technetium. It's mostly the answer with these things, mostly. Yeah.
Okay, thank you. So, Masema, in case you see these things, we bet our money on technician.
You can
Nice.
take it to.
Can take it to the bank. Okay, okay. Right. This is not a method of isotope labeling of compounds. Tagging reaction, fusion reaction, isotope exchange, isotope. Yeah, somebody's talking.
We can just add that iodine. The iodine is used for thyroid.
For their red tube, okay. Makes sense. Anybody else has an addition in this? Because like we're going to the radiopharmacy part.
Is there known for any particular thing like lungs or heart?
Let me see if I can get something about it.
I'm seeing an image, but I don't know.
I think we have ever done such kind of thing in class for isotopes and different uses. Just can't find where we did it.
Yeah, but I can't find that.
Mm. I can't find somebody is able to find it. Then maybe they're able to send it to the group.
I found an image. Let me share.
Okay.
Mumeona?
Ah, article about 60 edition therapy to prevent cancer. Heading that one. 131. Blockades brain tumors, cardiac, liver, and brain activity. Carbon 14. Study metabolic change in patients with diabetes, convenience, carbon 11. Carbon 54, so much and almost to extend. Sedentary classification. Seven. Yes.
Kuskipa.
Kuskipwa ama uniskipwa?
Uko mbali na mic.
Oh.
Aya,
Oh, well, now I'm clearer.
lisia.
So, I was just reading what Letting has sent to the group basically, so radioisotopes and some of the applications in medicine.
Ya selenium sijapata sana.
Ati nini? Yeah?
Selenium.
Selenium. Let me see, can get it. You can Google.
Ndio natafuta.
Or not tumeongezea more properties for technicians in radiopharmaceuticals. It's the most common atom used in radiopharmaceutical. Over 75% of all radiopharmaceuticals will include it. Some of their properties will it has a 6-hour half-life, a good half-life. The high abundance. It lacks particulate alpha and beta emissions. It also has a versatile chemistry that allows it to be chelated with a variety of compounds. This can be used. For selenium, hiyo sipati. Let's see ukipata you can share it with us also. So okay, move on to the next question. For that one, we'll go with TCO4. Okay. Dash is not a method of isotope labeling of compounds. So which are the methods of isotope labeling? Tagging, fission production, isotope exchange, isotope farming. I think the isotope farming. I think I don't know the answer for that one. Somebody says selenium is for pancreas imaging. Strontium SR. Hey, what is S? So SR is strontium. Strontium. Selenium is used for pancreas imaging. Okay, thank you, Sam and Felicia. So somebody says, I think isotope farming is not a method of isotope labeling. So all the others are forms of isotope labeling, tagging, fission, and isotope exchange. Okay. Okay.
Okay, I'm sure about A and C.
Yeah, I think so.
Sure about B. Not so sure, but I'm seeing a C if it was there, but not so sure. But A and C ones, I'm sure.
Isotope. Yeah, but my Google is broken. Has anybody found information on methods of isotope labeling?
Ndio kutafuta.
Ah, nice. Massive. Something has been sent to the group, and based on it, the answer is isotope farming. So what is isotope farming? Hey, natuma the references. So thanks for that, Dr. Sam. So we can see what tagging reaction is a method where a labeled precursor is reacted with an unlabeled compound to form a labeled product. For example, carbon 14 can be used to label organic compounds for photosynthesis or carboxylation. Fission production, that is a method where a radioactive isotope is produced by nuclear fission of a heavy element such as uranium or plutonium. The fission products can be separated and used as labels for various compounds. Okay. Isotope exchange is where a labeled atom or group exchanges with an unlabeled atom or group in a compound. It's like they're exchanging. It's like an elimination. Yeah, it's like a reactor. The exchange can be facilitated by catalyst, solvents, or temperature. Okay. Sawa, asanteni kwa hayo. So in case you ask methods of isotope labeling, there we go. Tagging reaction, fission reaction, isotope exchange. Isotope farming, however, is not. Yeah. Haya. So moving on, an isotope chain can detect which one of the following organisms. Kind of there.
Sikusikii.
Mistake.
Kwani
Napo-
sisikiki? Sina microphone.
Tafuta,
Like it's not
tafuta.
Kujia,
microphone.
kujia.
Well, usiniletee tu. Na sasa mnanisikia poa, so you should be shouting. My neighbors will come.
Utawafunza wakikuja. Eh,
You repeat. I also don't know what was said. So whoever said whatever it has been said, please repeat. And being kicked out.
ni wewe ndo ulikuwa unaongea. Anauliza isotope farming.
Hi, hello. Am I audible?
Yeah, can you repeat the last question, the one before? I think
Oh, it was asking something about acid fast staining.
what did you go for?
I was also not sure, but I'm thinking M tuberculosis, Mycobacter tuberculosis.
And could we share other acid fast staining microorganisms?
Yeah.
Do you know the drill?
Somebody, Dr. Munene has said Mycobacterium leprae.
Mhm.
Can you even ask about tests for these other microorganisms? Mycobacterium avium complex. So in short, you're talking about the Mycobacterium family.
Thank you.
Oh, it's only for those two. It's only for tuberculosis. Okay, somebody is saying they only use for tuberculosis and leprosy, but I've seen tumeongeza hiyo avium complex. Oh yeah. What about tests for Neisseria gonorrhea? I'm also not liking our questions. The kind of questions should be asked for. Next week,
Thanks. Hey,
we're on a spotlight. Let's get skip. Let me ask. There was this test we were doing for meningitis. Nimesahau. It has like 3 letters. J for the acronym. What was it called?
The one for pulling the leg.
Pardon,
No.
doctor.
No, okay, you can still say those ones for, you can still say the test for meningitis, okay. Those ones. Yes, correct test. That's what I was looking for. Yeah, okay. And which other one? Patrick, you brought that one up. What was that one called for pulling the leg? There was one for the leg and one for the neck. What were they called? You can't hear me? Hey, Bernard. Let me look for a microphone.
There was Koenigs and the other one was starting with a B. Bra something. I know Koenigs. Koenig and bra something. I can't remember that one. Bradzinski. Yes. Bradzinski. Yeah.
Okay, which one was for the leg? Which one was for the neck?
For the neck is the Bradzinski. For the leg is another one.
Okay, thank you.
Koenig.
Any other questions on this? Any other question? Any more additional information? I can keep them coming even as we move on. Let's go to the next question. Dash is used in the breath test for H. pylori infection.
Urea. Oh, urease.
Yeah, I agree with you. I wanted to make it by over there. That tested. Thanks for that. Also used with protease species. Okay. Need to remember the urease test can also be used for the protease species. Thank you. Which of the following genera are coliforms? Somebody start by telling us what's the coliform and then tell us which of the following genera are coliforms. Or just answer, or just say what you know. Coliform, which I have go. Hey, somebody has said that is bacteria always present in the gut. Okay. But if it's bacteria always found in the gut, yeah. So it's gram-negative and rod-shaped. So okay. So these are bacteria always found in the gut. They're gram-negative and rod-shaped. Found in the colon. They belong to the Enterobacteriaceae family, such as the E. coli. Okay. Feeling the gram-negative and rod-shaped is also important because the Clostridium difficile is also present in the gut. Non-spore forming. Okay. Right. Okay. So what shape is Clostridium difficile? Now we have established E. coli is gram-negative and it's rod-shaped. What about Clostridium? what it's a cocci. Gram positive or gram negative? It's a gram positive anaerobic cocci. Okay. Let's go on. Which of the following viruses has a segmented genome structure? We have the influenza, the polio, rabies, and the yellow fever. Dr. Munene says influenza. Who am I to differ? I don't know if the notes that have been written on these are correct. And I think the polio virus is non-enveloped, rabies enveloped, and the yellow fever virus is enveloped. And which ones were more susceptible? I don't know whether it goes for the viruses also to destruction or to what do you call something that can be destroyed by heat. Yes, envelopes are more susceptible. Why? Why? I hope you're typing. I think you're typing. Hi, if he thinks of the answer, we can be moving on. Which of the following viruses cause latent infection? Oh, all the following viruses cause latent infection, except herpes simplex, hepatitis B, cytomegalovirus, and varicella zoster virus. And he says, because you just need to destroy the envelope and the virus is dead. Hence why sanitizers worked against COVID. Yes. You just need to destroy the envelope. Dr. Nugal, you are correct. Do not second guess yourself. Okay. Yeah. So all the following can cause latent infections except I know herpes simplex latent infection. And that's about where my knowledge in virology goes. Okay, thanks, Tur. So latent infections are caused by the Epstein-Barr virus, the VZV virus, the herpes, and cytomegalovirus. Therefore, B. Okay, you said herpes and you said cytomegalovirus, and you have both of them. Okay, herpes. Oh, the VZV is the varicella zoster virus. Okay, so the answer is B, hepatitis B virus. Also HIV causes, yeah. Hepatitis B, we have the A, B, C, and they affect different, correct me if I'm wrong, again, my virology knowledge. They were affecting, they have different ways of catching the infection and the parts of the body they affect, right? So where does hepatitis B, I'm confusing. Let me just confirm before I confuse you all. I found it. We have hepatitis A that's transmitted via the orophycal route. or contaminated food. We have the hepatitis B, which is transmitted via blood to blood or through sexual intercourse. C, also blood, or the B, C, and D can be transmitted via blood to blood. A and E through the fecal oral route. And moving on. All the following done that. Which of the following organisms
There
cause
was something,
diseases? Uh-huh.
I think there was something about the hepatitis virus that causes some, I don't know, liver disease in pregnancy or something to do with pregnancy. Someone could tell us. Anyone with good memory on that?
Let's see. B. I'm seeing acute hepatitis B virus who pass the virus to their babies. All of different. C. So your question is on the one they get during pregnancy or the one they can pass to their babies. Which was it? What's the question?
Okay, I think it has been answered in the chat. I think
Oh,
it's hepatitis B.
yeah, dangerous. Hepatitis E is dangerous for pregnancy. Okay. Okay. Which of the following disease in humans through toxins? Protozoa, fungi, algae, arthropods.
Done. Janet.
Yes, yeah.
For just a quick one. For latent, it means it lays dormant, Ama.
Yeah. It means it can lay dormant.
Okay. Okay, thank you.
So which one causes disease in humans through toxins? Organisms. Yeah, that's the answer. I'm finding fungi. No, algae does have toxins. Arthropods, these are like animals. They don't have toxins. And some fungi. And don't fungi also have toxins? Okay, what is your thoughts on this? Because me, if I have been called this, I'll end up having toxins on each class. Because it's disease in humans through toxins. Okay, any thoughts on number 34? Okay, you can leave a star on it then. Don't all A, B, and C release toxins? I thought so. And if you ask me, even D will also have toxins. What are the animals under arthropods? Let me see. Millipedes, scorpions, crustaceans. Hey, even centipedes. Okay, I number 34. I guess you can leave a star on it. Huh? Bacteria and fungi. Okay. Yeah, but does that definition help us answer the question? We can leave number 34 for further studies and go as people B, fungi. Okay.
Yeah, actually for B, you can take an example of the aflatoxins from the Aspergillus species. These toxic mushrooms, they all produce toxic substances. So I think fungi is correct. For protozoa, we don't know any toxin from protozoa or algae or arthropods is a general term of which they don't really produce toxin. So
Of which they don't?
they don't really produce toxins, but it's actually a general term. Okay, yeah, they actually don't produce toxins. It's mostly venom in terms of from the arthropods. So I think this is actually the most suitable answer for us.
Okay. All right. So fungi it is. Somebody sees 35, answer 34. So aspergillus. Okay. Which of the following fungal species produces toxins that are carcinogenic? Coccidioides, Peptidos, Cytoplasma, Capsulae, Candida albicans. And Dr. Sunshine says aspergillus. So I learned. Okay. Any questions on 34 and 35? Any additions? Yeah.
So as an addition, Aspergillus
Mhm.
is actually a genus in the fungus domain. So basically, it actually cements the answer that we put for number 34.
Yeah, I'm saying somebody had said the two are related. Okay, the two answer each other. Right. All the following diseases are transmitted by insects. Obviously, testes, meningococcus, Diphtheria, caninum. And then you have the dyspepsia. Mhm. And our good host tells us it's like rickettsia. Because rickettsia is spread by is transmitted by, or rather mites, fleas, figures, mammals. What spreads what transmits it? So I'm finding it's transmitted to humans by the bite of infected ticks, mites, and by the feces of infected lice and fleas. So it is transmitted by fleas. Eight. Basically, they're all transmitted by fleas. But okay, but E. rickettsias seems to have several vectors. Oh, okay. So yes, Nepenthes is transmitted by fleas, mirroring typhus is transmitted by fleas. Syria, the picture.
So we have rickettsia that causes
Mhm.
Rocky Mountain spotted fever, and the vector is tick. Then there is Rickettsia typhi, and now the vector is rat flea. Then you have Rickettsia acari, and then now that's the vector is mite, and then now Rickettsia prowazekii, then the vector is louse.
Okay. So you see rickettsii, that's by a mite.
That's Rickettsia acari.
And this one, rickettsii.
Tick.
What?
Tick.
Ticks.
Are you saying a pig or a tick?
Tick.
Tick. Oh, okay, tick.
Yeah.
Thank you. Thank you for that, Dr. Asmahan. Sheila, you can go on. And more vectors in the group. So which of the following has do not eat? Sandfly, coxsackie, dengue, and rabies. How? Okay, so which is not a zoonotic disease? Pardon, the answer to question 36. The answer was the rickettsia rickettsii, because it's spread by ticks. And the answer for this one has been said is a Coxsackie virus. Coxsackie B, because all causes, what spreads, what transmits. So what transmit or spreading? I don't know. Rabies. And who also knows the rabies guidelines? What is it called? The treatment guidelines for rabies.
For rabies, I'm not so sure if the timeline is correct, but what I know is that there's a what is it? We call it post-exposure prophylaxis because there's no cure per se. So you'll given a shot, I think on day 0, day 3, day 7, day 14. Yeah, those 4 doses, whereby you're given some, it's a rabies vaccine. So you're given on day 0, 3, 7, and 14. If I'm not wrong, someone can just confirm that 0, 3, 7, and 14. We give you
sometimes, yeah, you're right, Dr. Gav. Sometimes I think there's 2 extra, 28 and 90.
now
90 days. Yeah.
According to my where I read, it had said that the 28th day thing was no longer recommended, but I don't know what is currently being practiced in the country.
Okay.
Yeah.
So the most important ones is day 0, day 3, day 14. That's 2 weeks later.
Day 7 and day 14.
See, also on that day, semester day 0.
Yeah, 0, 3, 7.
Day 0, 3, 7, 14.
Okay. All right. And which are the most common or obvious symptoms for rabies?
Hydrophobia.
Hydrophobia. That is when it gets very bad. No return. Okay. Which of the following is a common causative agent of boils? That one you can do another bonus point, eh? Staphylococcus, yeah. All the following epidemic prone diseases requires a single confirmed case for action threshold, except influenza, meningococcal meningitis, cholera, poliomyelitis.
Influenza. Yes, tutakuwa tumefanya kwa pepa ingine. I think ilikuwa influenza.
Okay. Chancroid is caused by Petro, hii inafaa kujua.
Ilikuwa
Chancroid,
treponema.
yeah. It's a common
Treponema pallidum.
symptom for
Syphilis.
You didn't know, right?
Yo, Chancroid, the Ducrey. Treponema.
Ducrey.
Yeah, treponema is a chancre.
Oh, it's a
Painless.
chancre.
Oh.
Right.
What's the difference?
Treponema,
What's the difference?
Yeah.
Difference ni?
One is cause in Haemophilus ducreyi causes a chancroid. I think the chancre, yeah, syphilis is just some painless chancre, but the chancroid, I'm not sure. I'm not sure. Perhaps Chai can tell.
Maybe the chancroid can be painful.
Chancres are painless. Chancroids are painful. Yeah, correct. Actually, ni Haemophilus.
Okay,
Alright.
thanks.
Thanks for the clarification.
You're
So
welcome.
chancre is syphilis and chancroid is ducreyi. Haya.
So, imepata hapa. So a chancre is one and chancroid are multiple.
So that's the difference.
Yeah. Edges of the chancre are regular. Higher. Chancres ooze serum and do not bleed easily, while chancroids ooze pus and bleed easily. So chancre doesn't ooze pus. Higher. For the what? Lymph, they're saying discrete, painless, and never suppurate. Suppurate, open fistula, and have sinus. I don't know what that means, but yeah. And it takes, for the chancre, it takes 3 weeks incubation. And for the chancroid, it's 4 to 7 days incubation. Yeah, that's it. And the chancroid forms an ulcer. Okay. Yeah, I think that's about it.
Okay.
Alafu
stage
on
of malaria.
rabies, it's 5 doses for guys who are post-exposure, in people who are not who have not been previously immunized. For guys who have been immunized, it's 2 doses on day 0 and 3. People who are not been immunized, it's 5 doses, day 0, 3, 7, 14, and 28. Yeah. And also for for post-exposure on people who have not been immunized, it's 5 doses. That's on day 0, 3, 7, 14, and 28. For people who have been immunized, it's just 2 doses, day 0 and 3, post-exposure. And pre-exposure, it's 4 doses, day 0, 7, and 21 or 28.
The stage of malaria parasite ingested by mosquitoes is called. That's the gametocyte. Have we done like, I don't know, a crash on the of the malaria, or have we discussed about malaria? Is that a yes? Well, basi tukaenda discuss the ones that have been written there, the trophozoite. At what stage does it become a trophozoite? At
Okay,
what stage does it
we can do the malarial cycle.
Yes.
So it's okay. It's basically caused by protozoa, right? It can either be Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, malaria, or Plasmodium novelsi. But Plasmodium novelsi is more of zoonotic. And malaria is the mode of transmission is like a bite from a female Anopheles mosquito. Do we need to know the signs and symptoms? Okay, just common one is fever, chills, abdominal pain, muscle pain, joint pains, and such. Plasmodium falciparum. When you're doing a diagnosis, you can see that it has histidine-rich protein 2, while the other plasmodiums have lactase dehydrogenase or aldolase. And for treatment, you give azimeter and lumefantrine, or you can give for pregnancy, quinine and dextrose is given just to ensure that the patient doesn't experience hypoglycemia. And second and third trimester, you can give artesunate with amodiquine. You should not give primaquine to any pregnant women because it is teratogenic. Okay, so for the malarial cycle, the infected mosquito is going to bite and leaves sporozytes. And then the sporozytes will move to the liver to form merozytes. And then from the liver, they are released into the red blood cells. So in the red blood cells, they'll convert merozytes to trophozytes, and then they'll keep on multiplying to form new merozytes. Now the merozoites are the ones that convert to gametocytes. So if you have malaria and another mosquito comes to bite you, they'll take the gametocytes, and the cycle proceeds. Repeats. So pregnant women should not take primaquine or tetracycline because it's teratogenic. First trimester, you can give quinine plus dextrose. Second and third trimester, you can give artesunate and plus amodiquine, or azimeter with lumefantrine. Yeah. Or that was just for Plasmodium falciparum. But if it's another like Plasmodium vivax ovale, and if it's not very complicated, you can give chloroquine. I don't know if anybody wants to add on anything, but that's all I have. Thank you.
Thank you. You've covered the entire life cycle.
It's okay. I just summarized it that the mosquito is going to give sporozytes that will go to the liver to form merozytes. And then the merozytes will be released and they'll invade the red blood cells to form more merozytes that are converted to trophozytes. And then the trophozytes will be converted to more merozytes. And then the merozytes become gametocytes. Then the mosquito ingests the gametocytes.
Okay, thank you for that, Shreena. Somebody is requesting we go back to number 39.
Yeah.
So, okay, what about it? All the following epidemic prone diseases require single confirmed case of AIDS. Like, what about number 39? Discuss further, explain further.
Janet, can you hear me without breaking?
Yeah, you okay?
The network issues, but it was more about the diseases that should be refined. A patient, even one patient with that disease, you need to report to the Ministry of Health, through various offices, but essentially they need to be reported to the government. Just polio, cholera, things such as guinea worm, such things. But influenza is something that happens in the community. It's the flu. We don't, as healthcare workers, we are not obligated to report every case of influenza we get.
Thank you for that, Dr. Then somebody has added on the malaria case. Relapse is caused by the PVVAX and Piovel. Recrudescence is caused by P. So what was that? It's caused by the P. falciparum. Thank you. We can move on. Any questions or addition for malaria? Feel free to text them on the group chat or in the group. The site of protein synthesis is, that's obvious, as the ribosome. The mitochondria is a powerhouse endoplasmic reticulum that is for assembling, and if it depends on which one it is. And the nucleus controls the functioning of the cell. Apoptosis occur only in neurons, is a programmed cell death, is only triggered by external stimuli, releases new cells. Apoptosis is a programmed cell death. And the other one, which is not programmed, is called necrosis or something like that. I don't know the name. Oh, no, I see you're adding more. Somebody has said falciparum is a black water fever and can also cause a cerebral malaria. Coxsackie spread by fecal oral roots. Nyaribo, do you mind talking more about the Coxsackie bee? Kindly. Okay, in her own time, she will.
Okay.
Uh-huh.
Coxsackie, we have Coxsackie A and B.
Mm-hmm.
And Coxsackie is a positive stranded RNA, and it's spread by the fecal oral route. That's all I know, actually.
That is more than enough. Somebody else who has something else to add on the Coxsackie A or B? I don't know what he has said. Tutafutiange atatuongezea. We can be moving on. Unless you want to talk about the others, just the sandfly, the dengue, and the rabies. We've talked about rabies. Have we spoken about the sandfly? Oh, that's not really a disease. What does it spread? What does it transmit?
Leishmania.
Leishmania. Did we have two forms of leishmania? I think we have two forms of leishmania, the cutaneous and the visceral leishmania.
And mucocutaneous.
And the mucocutaneous leishmania. Somebody has to give me more information on leishmania. Its treatment or its symptoms. Let me get this. For the mucocutaneous, you're giving sodium antimony gluconides. Amphotericin B can also be used for resistant or advanced cases. That is for the mucocutaneous. For the visceral, you give the liposomal amphotericin B. I don't know what you do for the cutaneous. Somebody else wants to add something about it? About leishmania, what you know about leishmaniasis? What time is it? Never mind. 4 minutes. We can be continuing on. Somebody has added paramomycin. That is for leishmania. So can also be treated with paramomycin and sodium gluconate. Yeah. And canine. Wait. So how many types of leishmania do we have there? We said there's the cutaneous, mucocutaneous, visceral, and we have the canine leishmaniasis. And the vector is N. infantum. You guys have a lot of information. This is beautiful. Somebody else has gone on, Dr. Betty has given us a different causative agents for the different types of leishmaniasis. For the cutaneous, it's caused by the L major, the visceral by Donovani, and the mucocutaneous one by the Brasiliensis. And which one was more, do I say serious? I think the word is serious or fatal or something like that. I think the cutaneous, the visceral, the mucocutaneous colors are. The Kalaza was one which was fatal, if left, which is, the Kalaza is just it's the visceral, right? The Kalaza is the visceral leishmaniasis. Correct me if I am wrong. My name is, guys, is it the same? Yes, it is. Okay. Trina has sent us more information about PPB. What is it? It's a common virus that lives in the digestive tract. It has 6 subtypes. I'm guessing it's A, B, C, D, E. What can it cause? It causes, if the virus is typically not serious, it's rare and can cause heart inflammation, chest and abdominal muscle spasm, diabetes, and meningitis. It is transmitted through the fecal-oral route and secretions of infected people. You can prevent it by washing hands frequently.
Is it RNA or DNA virus, Janet?
Somebody said, who was Nyaribo? You said it was
Somebody said positive stranded RNA.
There's a positive stranded RNA virus. Do we stop there? Hata hiyo bado hamjibu. I am not insisting. Orea fever for sunflower. Hey, I didn't know about Orea fever. Okay, Orea fever for sandflies. I will expound more. Okay, give us a 1 minute crash on Orea fever, if you don't mind. Guys, since you make exclamation, I know you also learned this today. What is Orea fever?
It's a fever.
My Google is asking if it's a real thing. You see, some of these things. It's also, oh, it's a carrier's disease. Okay. Is it, wait, you guys, I don't know which books you're reading, but you need to give me those books also. Ah,
Anyway, we can stop there to continue at 8. 2 hour break.
yes. Adios.